Hi students, myself Satish Kumar Konidala, Assistant Professor, Faculty of Pharmacy, Vignan's Foundation for Science, Technology and Research, Guntur. In this video, we are going to discuss about carbohydrate metabolism. As we know that, so there are different types of biological molecules are available in the living system. So there are in that there are some major molecules and some minor molecules. So these major molecules are composed to form the different types of macromolecules, cells, tissues, organisms, and finally one organism. Organs and organism. Okay, so these molecules sometimes they are subjected to metabolism to perform various biological reactions. Understood? So to perform the various biological reactions, these biological reactions are essential for livelihood of the organism or living of organism. So the biological molecules, biomolecules subjected to metabolism. So this metabolism is nothing but they are performing different biological reactions, and these reactions, these biological reactions are essential for our living systems. Okay, so for that reason they may subjected to metabolism. So let us see different types of metabolisms, different types of biological molecules, how they are subjected to metabolism. In that, first one is a carbohydrate metabolism. So in this video we are going to discuss about the carbohydrate metabolism, how the carbohydrate is subjected to metabolism in different pathways. So what are the different pathways of carbohydrate metabolism, and what is the need of, or what is the requirement of carbohydrate metabolism, and what are the disorders during the metabolism of or related to the carbohydrate metabolism. Coming to the learning outcome of this video includes, by watching this video, student could understand the metabolism of carbohydrates in physiological and pathological conditions. Okay, so they can understood the metabolism, how the carbohydrates are subjected to metabolism, and what is the role of this metabolism of carbohydrate in physiological and pathological conditions. And by watching this video, student can able to apply the knowledge of this carbohydrate metabolism to estimate various biochemical parameters in physiological systems. Okay, so these are the outcomes of this video. Particularly in this video, we are going to discuss about the some parts of carbohydrate metabolism, that includes introduction, glycolysis, and citric acid cycle. Let us see one by one. Coming to the introduction, so we know that carbohydrates are the major source of energy for the living system, and these carbohydrates are the first cellular constituents that can be synthesized by the green plants during photosynthesis from carbon dioxide and water by utilization of the light. So by utilizing the light, the green plants are synthesizing the carbohydrates as the first cellular constituent from carbon dioxide and water. Okay, So we can consider the light is the ultimate source of energy for all the biological systems. So without the light, we cannot able to produce the carbohydrate by using the carbon dioxide and water. Okay. So for that reason, light is considered as the ultimate source of energy for all the biological process. Okay. And in case of monohydrate, in case of monosaccharide, there is a glucose. So the glucose is the one of the monosaccharide, and that is central molecule in all carbohydrate metabolism. So wherever the carbohydrate is subjected to metabolism, so there we can consider or we can concentrate on the glucose. Okay. And glucose is utilized as a source of energy, and it is synthesized from the non-carbohydrate precursors and stored in the form of glycogen. Okay. So when there is a need of energy and that stored glycogen is released into glucose to meet the requirement. Okay. So first the energy is in the form of glucose, and this glucose is synthesized and it is stored in the different tissues like adipose tissue. It is stored as the glycogen, and this glycogen produces the glucose when there is a energy needs are occur. Okay. And other monosaccharides which are important in carbohydrate metabolism are fructose, glucose and mannose. And here we can see the different types of carbohydrates, how they are subjected to metabolism. So in case of starch, starch is subjected to metabolism by the enzyme. Different types of enzymes are involved to metabolize the particular type of carbohydrate into their precursor, their metabolites. So in case of starch, starch is metabolized by the enzyme that is a salivary amylase. It can be converted into maltose and dextrins. And again the starch is metabolized by the pancreatic amylase. It can be converted into maltose and isomaltose. And maltose is converted into glucose by the action of maltase enzyme. And isomaltase is converted into glucose by the action of isomaltase. Lactose will be converted into glucose and galactose by the action of lactose. Sucrose is converted into glucose and fructose by the action of sucrase. Okay. So these are the some example how the dietary carbohydrates will be converted or will be metabolized into their metabolic products. And so the carbohydrate is subjected to metabolism, and then it can produce the glucose, and that glucose, it is considered as a monosaccharide. How it is going to be absorbed? So there are three ways to absorb the monosaccharides. First one is a simple diffusion. Simple diffusion, that means according to the concentration gradient. So concentration gradient means, for example, in our intestine high concentration of monosaccharides are present, and so this is the barrier between the intestine and blood circulation. So if the blood circulation has low concentration of monosaccharide, then the diffusion takes place from high concentration to low concentration. So there is a simple diffusion process. So examples, fructose and pentoses are subjected to simple diffusion process. And facilitated transport. Second one is a facilitated transport. For example, in case of glucose, example is GLUT5. So GLUT5 is the protein which can facilitate the fructose transport from intestine to blood. So here the protein GL25 is the one of the protein that can facilitate the transport of fructose from intestine to the blood. So that means the fructose will carried by this protein, and it can be transferred from intestine to the blood. Okay, and next one is active transport. Active transport. So active transport means there is a utilization of energy. Okay, so example is sodium glucose transporter. So 1 sodium glucose transporter 1. So this is the one of the glucose transporter. So it can facilitate the reabsorption of glucose in renal system. Understood? So it can facilitate the renal glucose reabsorption process. So sodium glucose transporter SGLT1. So it is a glucose transporter, so found in the intestinal mucosa. So it can bind the glucose, or it can carry the glucose, and it can involve in the active transport. Active transport means maybe from low concentration of the glucose to high concentration of glucose through some carrier. So that carrier is nothing but SGLT1 for the glucose. Okay, so this is majorly or generally involved in the case of renal reabsorption of glucose. Renal reabsorption of glucose. So these are the different ways of absorption of the glucose or monosaccharides. And let us see what will happen after absorbing the monosaccharides or sugars. So we already seen that there are different ways to absorb the monosaccharides from dietary source, that is from intestine to the blood. So after absorption, what will happen? So after absorption, the fructose or galactose, so they are stored in the form of fructose or galactose, so that are transported to the liver, and this liver will break down these molecules fructose and galactose into glucose, and this glucose which is formed from the liver is taken up by the tissues for their need or to utilize for their daily need. Okay, and coming to the pathways of glucose utilization, how the glucose is utilized by the tissues. Okay, so first one is the glycolysis. So there are different pathways for glucose utilizations. In that, first one is the oxidation pathways. So the oxidation pathways are nothing but they can produce the energy. So the pathways based upon the oxidation can produce the energy. So in that the glucose is actively utilized, and the glucose can provide the other compounds like carbohydrates like fructose, galactose, and pentoses, glyceraldehyde, and it is also produce the glycerol 3 phosphate, so like triglycerol and phospholipids, and can produce the acetyl CoA, so which can convert it into cholesterol and fatty acids. From this acetyl CoA there is a production of cholesterol and fatty acid, and non-essential amino acids are also produced by using the glucose. So that means in all the pathways, so that means these are some types of pathways, so that can able to produce the different products. In that pathways, the glucose is actively utilized, and the glucose can be stored. So it can be stored as glycogen in liver and triglycerol in the adipose tissue. So the glucose can be stored in the form of glycogen in the liver and triglycerol in the adipose tissue, and it is also excreted. So and excess amount of glucose is excreted in urine. Okay, this is the pathways in different. So this is the fate of glucose, particularly glucose, how it is subjected to different type. That means after absorption of the glucose or sugars, so it is converted into glucose, and this glucose is subjected to uptake by the tissues. So during this uptake or during the utilization of this glucose, it is subjected to different types of oxidation process, or it can be used for the providing or producing of compounds like carbohydrates, glycerol 3-phosphate, acetyl CoA, and non-essential amino acids. And these compounds again they are converted into other corresponding compounds, and some of the glucose may be stored in the form of glycogen and triglyceride in liver and adipose tissue respectively, and some excess amount of glucose is excreted in the urine. So this is the fate of glucose when it is entered into the biological system. And coming to the oxidation of glucose. So in pathways what we discussed in the previous slide. So first one is the oxidation of glucose. So the oxidation of glucose is explained in two pathways. First one is a major pathway and second one is a minor pathway. Major pathways includes glycolysis and Krebs cycle, or it is also called as citric acid cycle. So major pathways, so they can produce the energy. Major pathways are producing the energy, and minor pathways are utilized for the synthesis of other derivatives. And in case of energy production, there are two pathways are there. First one is a glycolysis and second one is a Krebs cycle, or it is a citric acid cycle. Glucolysis produce the pyruvate, and so in case of glycolysis, it produces the pyruvate under aerobic condition and can produce the lactate and anaerobic condition. So during the glycolysis process, there is a two metabolites are produced. First one is the pyruvate and lactate. Pyruvate is produced under aerobic condition. Aerobic means in presence of oxygen. Lactate is produced under anaerobic condition. So that is without the oxygen. Okay. And Krebs cycle. So it is another pathway, comes under the major pathways of oxidation of the glucose. So this Krebs cycle under aerobic condition, the pyruvate, the produced pyruvate in the glycolysis is converted into active acetate for oxidation. Okay. So the pyruvate which is produced in the glycolysis is subjected to oxidation under aerobic condition, that is nothing but the Krebs cycle. Okay. And coming to the minor pathways, so they are generally meant for the synthesis of other derivatives. So in that, first one is a hexose monophosphate pathway, or HMP pathway, or it is also called as HMP shunt. So it is used for the production of pentoses and NADPH. The hexose monophosphate pathway is, or it can produce the pentoses and NADPH. And uric acid pathway, it can produce the uronic acids. Uronic acid pathway can produce the uronic acids. So these are the pathways involved in the oxidation of glucose. So oxidation of glucose are of two types. Oxidation of glucose is classified into oxidation of glucose can be carried out by different pathways. In that they are classified into major pathways, minor pathways. In that major pathways, the first one is a glycolysis and second one is a Krebs cycle. And major pathways are meant for energy production, and minor pathways are meant for the synthesis of other derivatives. In that, first one is a hexose monophosphate pathway and second one is a uronic acid pathway. Okay. First we will discuss about the glycolysis. So glycolysis is the major pathway of oxidation of the glucose. So it can able to produce the energy. Okay. So when the glucose is utilized by the glycolysis, then it can produce the energy. So the glycolysis is nothing but oxidation of glucose to pyruvate in presence of oxygen, or it can produce the lactate in the absence of oxygen. So presence of oxygen is nothing but aerobic, and absence of oxygen is nothing but anaerobic. So the glucose, when it is subjected to oxidation in presence of oxygen, then it can produce the pyruvate, and glucose subjected to oxidation in the absence of oxygen, then it can produce the lactate. So these two are happen in case of glycolysis. So the site of glycolysis pathway is nothing but cytosol. Cytosol means, so cytosol means there is a cell. In that cell, the different cell organelles are there, and inside the cell, all the cell organelles are floated in a cytosol. So this is called as the cytosol. The fluid which is present inside the cell is called as the cytosol. The site of glycolysis pathway takes place in cytosol of the cells. And coming to the steps involved in this glycolysis are, so actually there are three, but here they mentioned two. So first one is a energy utilization. The first phase of the glycolysis is nothing but energy utilization, and second one they mentioned is energy recovery phase, energy production. Energy recovery means energy production. But between these two there is another phase, that is a splitting phase. So after energy utilization phase, there is a splitting phase is there. So that is a second phase of the glycolysis. So after the splitting phase, the third phase is nothing but energy recovery phase or energy production phase. So during the first phase, that is the energy utilization phase, glucose is cleaved. So first the glucose will be converted into fructose 6 phosphate, and that one be that is a 6 membered carbon molecule, and that can be cleaved into 2 molecules. Okay, 2 molecules. So each molecule consisting of 3 carbon atoms in their structure. The 6 membered carbon molecule will be cleaved or break down into 3 numbered carbon molecules. So like glyceraldehyde 3 phosphate, so glyceraldehyde 3 phosphate has 3 carbons in their structure. Okay, the 6 membered what is that, fructose 6 phosphate, it is a 6 membered ring molecule that will be converted into, sorry, not 6 membered, 6 carbon molecule that can be converted into glyceraldehyde 3 phosphate. This is the 3 carbon molecule. Okay, so this is the phase. So it can consumes 2 molecules of ATP. So the first phase consumes 2 molecules of ATP. That means it is the energy utilization phase. Energy utilization phase. And second phase is the energy recovery phase. That means energy production phase. So the 2 molecules of glyceraldehyde. So here I said that 1 molecule of fructose 6 phosphate will be converted into glyceraldehyde 3 phosphate of 2 molecules. And these 2 molecules of glyceraldehyde 3 phosphate are converted into pyruvate under aerobic state or aerobic condition. So by the generation of 10 ATPs. So it is subjected to different conversions. So the 2 molecules will undergoes different biological reactions. They can produce the pyruvate. Along with this pyruvate, they can produce the 10 ATPs. And or in case of anaerobic condition, it can be converted to lactate. By the or along with this lactate, it can produce the 4 ATPs. 4 ATPs. All reactions are, all intermediate reactions which are involved in the glycolysis are reversible in nature, except these intermediates, that is GK, PK, PFK and PK. So I will explain all these in detail. So first one. So here the glycolysis. So glycolysis is the Greek word which is derived from the two words, that is a glyco means sweet or sugar. Glyco means sweet or sugar, so that is nothing but the glucose. Lysis, lysis means dissolution. Lysis means dissolution. So this glycolysis pathway is the one of the major pathway that can utilize the glucose, and this pathway is also called as EM pathway. EM pathway. So it is fully elucidated in the year of 1940. EM means embedded Meyer-Hof pathway. So it is also called as EM pathway or embedded Meyer-Hof pathway. So the pathway is fully elucidated in 1940. Okay, and coming to the definition, glycolysis is nothing but a sequence of reactions or sequence of events that can convert the glucose into pyruvate or lactate by the production of energy, or it can convert the glucose into. So there are different sequence of events that can convert glucose into pyruvate or lactate along with the production of ATP. So when the pyruvate is produced, when there is a utilization of oxygen. When the lactate is produced, when there is no utilization of oxygen. So due to when the absence of oxygen is there, then the glucose will be converted into lactate in glycolysis. Okay. And coming to some silent features of this glycolysis, as we know that glycolysis is happened in the cytosol. That means the enzymes hexokinase and fructokinases are present in the cytosol of the cell, so that the glycolysis has happened in the cytosol of the cell. And in the presence of oxygen, the glycolysis pathway will convert the glucose into pyruvate, and in the absence of oxygen, the glucose is converted into lactose. And this glycolysis is major pathway that can produce energy from the glucose, and it is also important pathway for the brain. So during the glucose utilization by the brain, glycolysis is one of the major pathway to utilize the glucose by the brain. Okay, so these are some silent features of glycolysis. And coming to the sequence of events involved in the glycolysis, so the glycolysis is classified into three stages. First one is a energy utilization stage, splitting stage, second one is a splitting stage, and third one is a energy recovery stage or energy production stage. So in first stage, that is a energy utilization stage, so there are some sequence of events are there. First one is glucose will be converted into glucose 6-phosphate by the action of hexokinase. So there are two types of kinases are involved in the glycolysis. First one is a hexokinase and second one is a glucokinase. So hexokinase is present in all types of cells, but glucokinase is present in the liver cells only. Okay, and this hexokinase has the more affinity towards the glucose, but glucokinase has the less affinity towards the glucose. So for that reason, in low concentration of glucose, in that situation also, hexokinase will convert the glucose into glucose 6-phosphate. But whereas in low concentration, glucokinase cannot convert the glucose to glucose 6-phosphate, but it can convert the glucose in high concentration, or when the glucose is present in high concentration. For example, in case of after meal, so we are having high concentration of glucose in the blood, that means more than 100 mg per dl. So in that situations, glucokinase will act on the glucose and it will be converted to glucose 6-phosphate. Okay, so that is a it depends upon the affinity and place of their conversion, or place of the glycolysis happen. So based upon the affinity of the kinases towards the glucose and site of action and type of these kinases will act on the glucose to convert glucose 6-phosphate. Okay, so that is a one concept. And coming to the sequence of events in the first stage, so first sequence of event is nothing but glucose will be converted into glucose 6-phosphate by hexokinase, by the action of hexokinase, by utilization of ATP, 1 ATP here. Okay, and after that glucose 6-phosphate will be converted into glucose 6-phosphate will be converted into fructose 6-phosphate by the action of phosphoglucoisomerase. So this is the isomerization of glucose 6-phosphate to fructose 6-phosphate. So the 6-membered ring will be converted into 5-membered ring here. So that is isomerization. So glucose 6-phosphate will be converted to fructose 6-phosphate by the action of phosphoglucoisomerase. So after that, this fructose 6-phosphate will be converted to fructose 1,6 bisphosphate. Fructose fructose 6-phosphate will be converted to fructose 1,6 bisphosphate. So this will be happened by the action of phosphoglucokinase. So this will be happened, or this will be converted. So the fructose 6-phosphate to fructose 1,6 bisphosphate will be fructose 6-phosphate is converted to fructose 1,6 bisphosphate by the action of phosphofructokinase by utilization of ATP, 1 ATP here. So the total energy utilization stage will utilize 2 ATPs. So the total first stage of glycolysis, that is a energy utilization stage. In that state, there are 2 ATPs are utilized. So one is at conversion of glucose to glucose 6-phosphate. After that, another one is fructose 6-phosphate to fructose 1,6 bisphosphate. Okay, and after the formation of fructose 1,6 bisphosphate, it will be split into 2 molecules. And here this is a 6-membered carbon molecule, and the splitted molecules are 3-membered carbon molecules. So the 3-membered carbon molecules are nothing but, first one is a glyceraldehyde 3-phosphate, and second one is dihydroxyacetone phosphate. So these two are reversible in nature. So that means that the dihydroxyacetone phosphate will be converted into again glyceraldehyde 3-phosphate by the action of tyrose phosphate isomerase. So these two are reversible in nature. So glyceraldehyde 3-phosphate and dihydroxyacetone phosphate are reversible in nature. So here, so there are some reversible reactions. So most of the glycolysis events are reversible in nature, except the glucose to glucose 6-phosphate and fructose 6-phosphate to fructose 1,6 bisphosphate. Okay, so these two are irreversible, and this conversion of glucose 6 phosphate to fructose 6 phosphate is reversible, and again glucose again conversion of dihydroxyacetone phosphate to glyceraldehyde 3 phosphate. So these two are reversible reaction. Okay, so the splitting process or the splitting stage of glycolysis is nothing but conversion of fructose 1,6 bisphosphate to glyceraldehyde 3 phosphate and dihydroxyacetone phosphate. So after forming these two 3-membered carbon molecules, so they can be converted into 2 glyceraldehyde 3 phosphate by the action of tyrose phosphate isomerase. So that means one glyceraldehyde 3 phosphate is formed from the fructose 1,6 bisphosphate, and another one is the dihydroxyacetone phosphate, and this dihydroxyacetone phosphate will be converted to again glyceraldehyde 3 phosphate by the action of tyrose phosphate isomerase. Tyrose phosphate isomerase. So there are two glyceraldehyde 3 phosphate molecules are produced from the fructose 1,6 bisphosphate. Okay, so that is a splitting stage, and next one is a energy production stage. So third stage of glycolysis is nothing but energy production stage. So from the fructose 1,6 bisphosphate, we are getting 2 glyceraldehyde 3 phosphate, and this 2 glyceraldehyde 3 phosphate will converted to 1,3 bisphosphoglycerate. 1,3 bisphosphoglycerate. So during this reaction, 1,3 bisphosphoglycerate by the action of glyceraldehyde phosphate dehydrogenase. So glyceraldehyde phosphate dehydrogenase, this is the enzyme, will convert the glyceraldehyde 3 phosphate to 1,3 bisphosphoglycerate. So during this reaction, there is a liberation of NADH plus H plus from the NAD. Okay, so this liberated NADH is actively utilized in the electron transport chain, and when it is entered into the electron transport chain, then it can produce the 10 ATPs. 10 ATP. Okay, so the conversion of glyceraldehyde 3 phosphate to 1,3 bisphosphoglycerate is the most important step in the glycolysis. So from this we are getting some ATPs are produced when the NADH is liberated during this reaction, and it is involved in the electron transport chain. Thereby it can produce the 10 ATPs. Next, the 1,3 bisphosphoglycerate is converted into 3-phosphoglycerate, and here it can utilize 1 ADP and can produce the 1 ATP. So this reaction is catalyzed by the phosphoglycerate kinase. So the enzyme which can catalyze the conversion of 1,3 bisphosphoglycerate to 3-phosphoglycerate is nothing but phosphoglycerate kinase. So during this reaction, 1 ATP is produced. So after formation of 3-phosphoglycerate, so it will be converted into 2-phosphoglycerate by the action of phosphoglyceromutase. So the enzyme will involved in the conversion of 3-phosphoglycerate to 2-phosphoglycerate is nothing but phosphoglyceromutase. So it will convert the 3-phosphoglycerate to 2-phosphoglycerate. So then the 2-phosphoglycerate will convert it to phosphoenol pyruvate. It is one of the high energy compound, and by the loss of water molecule, the 2-phosphoglycerate is converted to phosphoenol pyruvate. So this reaction is catalyzed by the enzyme enolase. The enzyme which can catalyze the conversion of 2-phosphoglycerate to phosphoenol pyruvate. Phosphoenol pyruvate is nothing but enolase. Enolase is the enzyme that can catalyze the 2-phosphoglycerate to phosphoenol pyruvate. Okay, and this phosphoenol pyruvate will be converted to pyruvate by the action of pyruvate kinase. Pyruvate kinase. So here also 1 ATP is produced. 1 ATP is produced. So here, so total 2 ATPs are produced from the glyceraldehyde 3-phosphate. So one is at from conversion of 1,3 bis phosphoglycerate to 3-phosphoglycerate. So here it is a 1 ATP, and here also 1 ATP. So these two reactions are example for the substrate phosphorylations. So without the involvement of electron transport chain, directly from the substrate there is a ATP is produced. So for that reason these two reactions are considered as the example for substrate phosphorylations. So that is nothing but conversion of 1,3 bis phosphoglycerate to 3-phosphoglycerate by the action of phosphoglycerate kinase, and conversion of phosphoenol pyruvate to pyruvate by the action of pyruvate kinase. So these two reactions are example for substrate phosphorylation. So these are the two examples for substrate phosphorylation. That means they are directly, so they can directly produce the ATP from the substrate. So the enzymes can, so by the due to the action of enzymes on the substrate, they are directly producing the ATPs. So for that reason it is considered the substrate phosphorylations. Okay, and total how many ATPs are produced during this one? So 10 ATPs are produced when the NADH is produced in this step, and this NADH is enter into the electron transport chain. Enter into the electron transport chain. So there it can produce 10 ATPs, and after that we can see 1 ATP is produced here and 1 ATP is produced here. So total 12 ATPs are produced in the glycolysis. Okay. So this is about the steps involved in glycolysis, and there is another glycolysis process, that is anaerobic glycolysis. So in that case there is a production of lactate. So in the absence of oxygen, the phosphoenol pyruvate is converted into ketoenol pyruvate, and that ketoenol pyruvate by the action of lactate dehydrogenase, so that can be converted into lactate. So in that process there is only 4 ATPs are produced. So in the case of glycolysis, in anaerobic condition it can produce the 4 ATPs. Okay. So this is about the glycolysis. In next class we are going to discuss about the Krebs cycle, that means the conversion of pyruvate to, so how the pyruvate is involved in the glycolysis process and how it is producing the ATPs. Okay. So the conversion of pyruvate to ATP, so that is nothing but the Krebs cycle or citric acid cycle. We will discuss that cycle in the next class. We will discuss that citric acid cycle in the next video. Okay. Thank you. Thank you.